Assalamualaikum. My name is Manzura and the topic of my presentation for second lab is anti-inflammatory effect. So let's start with the introduction. What is inflammation? Inflammation is the natural protective response of the body to infection or injury. Inflammation is majorly marked by primary five primary signs of inflammation, which are pain, heat, redness, sweating, and eventual loss of function. Inflammation is a process which helps to maintain the tissue immune homeostasis under stress conditions. There are different types of inflammatory response, and the type of inflammatory response depends on the nature of the initial stimulus and its location in the body. And although there are different types of inflammatory responses, all of these have a common pathway, which consists of the stages such as the recognition of cell surface protein receptors of detrimental stimuli, and then the activation of inflammatory pathways, and then the release of inflammatory markers, and then the recruitment of inflammatory cells, and then the eventual effect on target tissues. The subtopic of my presentation is evaluation of anti-inflammatory effect of different substances by the pleurisy model. In the research paper that I've selected, uh, the research paper focuses on the anti-inflammatory effect and anti-inflammatory properties of butylenic acid and xylopic acid in the carrageenan-induced pleurisy model of lung inflammation in mice. So the type of inflammation we'll be looking at is lung inflammation. In this type of model, the carrageenan-induced pleurisy model is considered an excellent acute inflammatory model, in which the leukocyte migration and other relevant biochemical parameters involved in the inflammatory responses with the lungs can be quantified easily in the pleural exudates obtained. In this model of inflammation, different vascular events lead to vasodilation, which lead to increase in vascular permeability and cellular events. And because of this, the inflammatory cells moved by chemotaxis get activated and then phagocytize the offending materials. There is evidence that reactive oxidative species play an important role in the pathogenesis of inflammatory disorders. The carrageenan used in this experiment causes a shift in the balance in the level of the free radicals produced. So this can lead to the starting or the initiation of cell damage and tissue damage. That agents and it has been recorded that the agents which reduce inflammation will also reduce the oxidative stress. In this study, the fruits of Xylopia ethiopica, which is a local spice found in Ghana and different West African countries, which is usually used to treat arthritis and other inflammatory conditions, are used. And from this plant, the two components, xylopic acid and butylenic acid, have been isolated. And these two chemicals will be used as the anti-inflammatory agents against which we will see the anti-inflammatory effect. The materials and methods used in the study, mainly the methods used will be the isolation of the compounds from the plant material, that is the isolation of butylenic acid and xylopic acid from the uh, plant mentioned previously. And the drugs and chemical used will be diclofenac sodium, uh, which will be used as a standard anti-inflammatory drug, against which the activity of the xylopic acid and butylenic acid will be assessed. And then hydrogen peroxide, chloroform, bleached acetic acid, etc. And then carrageenan sodium. The compounds under study, the test compounds, butylenic acid and xylopic acids, will be administered orally. Uh, to the will be administered orally to the mice with clean 80 in distilled water acting as a vehicle. The animals used will be mice, and for the carrageenan-induced pleurisy, the mice will be treated with test compounds or solid. Either the test mice will be treated with the test compounds, and the control mice will be treated with only the vehicle 1 hour before injection of pleural inflammation. Then the pleurisy will be induced by injection of carrageenan via the intrapleural route on the right side of the chest. Uh, After 4 hours, the mice will be sacrificed by overdose of inhalation of diethyl ether. Chest cavity will be open and the pleural cavity will be assessed. It will be rinsed 1 milliliter of normal saline solution, and the resulting exudate from the pleural cavity will be aspirated into a different vehicle, and the total volume will be reduced into uh, an instrument, the Eppendorf tubes. Then these exudate, which were stored into the different apparatus, will be refrigerated at the given temperature, and then the lung tissue will also be isolated and then fixed in formaldehyde solution for histological examination, as histological examination is necessary to obtain the data we need for complete evaluation of the anti-inflammatory effect. The animal groups used will be those mice which are given the carrageenan but not the test compounds. Uh, then group two will be those which are not given anything, neither carrageenan nor the test substances. They are only administered with the 2 in 80 in distilled water. Then the next group will be only administered xylopic acid, and then the next will be only administered butyric acid, and then the last group will be administered with diglycine, the standard and inflammatory drug against which the activity of the different compounds are being assessed. Uh, then after the test was conducted in the study, the results show on the evaluation of the lung tissue histopathology that xylopic acid was shown to protect against the carrageenan-induced tissue damage compared to the tissue uh, was isolated from those mice which were treated only with the solid. And then increase it was also seen that increasing the dose concentrations increase the protective function against tissue damage. And the protective ability of butylenic acid was dose dependent, meaning that by increasing the dose, the anti-inflammatory activity of butylenic acid increased. The data obtained in this study shows that acute administration of xylopic acid or butylenic acid reduces neutrophil migration into the pleural space of mice 4 hours after administration of carrageenan. The inhibition of cell influx into the pleural cavity was associated with the marked reduction in oxidative stress markers and in inhibition of antioxidative enzymes. Pleurisy-induced with carrageenan in movements is an important marker to measure the inflammatory effects of compounds and isobates and the possible effects on inflammatory mediators in vivo. The lung pathology supports the anti-inflammatory activity of xylopic acid and butyric acid. In conclusion, the study provides evidence that the pre-treatment of mice with xylopic acid or butyrenic acid helps the development of helps in alleviating the development of carrageenan-induced pleurisy, and it also reduces oxidative stress, and it also reduces neutrophil infiltration into the lung, which will lead to a decrease in lung injury caused by injection of carrageenan. So that's all for my presentation. Thank you.